Good afternoon. My name is Jed Fahey. It's my pleasure to be here. Nicole has asked me to read off a few organizational details, and then the normal suspects are not here, John Grootman and Norma and Kala. So they've asked me to introduce myself. So I will introduce myself in a minute. But first, organizationally, welcome to the Maryland Cigarette Restitution Fund and Sidney Kimmel Cancer sponsored Lunch, Learn, Link seminar series. Sorry, I have to read this because I have scheduling conflicts and can rarely make it myself. It is a pleasure to be here. I've certainly attended some of them over the years. Would like to welcome the viewing audience from the Maryland Department of Health and Mental Hygiene, Howard University, the University of Maryland, and others. I'd like to remind everybody that this is being broadcast and recorded so that their colleagues can view it online. And I'd like to remind the in-house audience to obtain a ticket for lunch. Lunch is in the gallery just to the left. Left? Yeah, your left as you are looking up at the stage, I guess. And then right after leaving the auditorium. So make sure you get a lunch ticket from Nicole. Since no one's going to introduce me, welcome, Jed, to this lecture. I sort of hate introducing myself, but let me see if I can do it in 1 minute. I didn't know how many students would be here, so I was thinking maybe just telling you about my tortured path to get here might be as entertaining as anything. So I actually got my undergraduate degree at Johns Hopkins University up the road a long, long time ago, and then got a master's degree in botany, in algal physiology or marine botany. And at the time I was in a department at College Park that was heavily funded by NASA. We were very excited about feeding astronauts microalgae that were grown in their own pee. And so I enjoyed those sorts of experiments. Worked with spirulina, which is a blue-green alga that was just becoming known at the time and is grown in alkaline lakes like Lake Chad, Lake Titicaca, Lake Texcoco, in the tropics primarily. And I think it was that experience that got me interested in public health, an interest that I had to subvert for 15 years or so, because I then went into the biotech industry and worked in the biotech industry for about 15 years before coming to Hopkins in 1993. I was finally able to indulge my interest in public health and got a doctorate while I was working full-time here, worked with Ben Caballero in international health, and got my doctorate while I was working here. So I've had a sort of odd, irregular, unusual path here, but I've been here since 1993, long, long time. I was recruited here by Paul Talalay, who has been here for a very long time, and we sort of regard him as the grandfather or the godfather of this field of chemoprotection that I'll be talking about. And I'll refer to him a few times, but certainly he's introduced a lot of the seminal ideas and has been for many years a big part of it. So I put my email down there. I have a slide at the end with my email and the address for our center's site, the Chemoprotection Center. If I say things that sound like you want to follow up on them, I'd encourage you to go to that site. We don't have a professional IT person keeping it up. It's those of us in the lab, so it doesn't get attended to too regularly, but we at least post links to papers as often as we think of it. And it gives you an overview of what we're doing. So with that, I'd like to talk to you today about phytochemicals in line with the title. I sort of have to give you some definitions to start off. Phytochemicals, phytopharmacology. We've coined the term green prevention, which I'll talk about, but mainly I want to get to some discussion of mechanism-based or mechanism-driven clinical studies utilizing sulforaphane, the active ingredient from broccoli. And we'll talk about both of them and ad nauseum, and then you'll go out and get greasy pizza and look for the broccoli on it, I hope. for lunch. I'm sorry, Nicole, is it greasy pizza or is it delicious pizza? She's probably adding broccoli to it right now. So what are phytochemicals? There are various definitions. This is mine, and it's that they are non-nutritive compounds. In other words, compounds that are not required for your survival. They're plant chemicals. They confer health benefits on humans. By the way, they also confer health benefits on plants, the plants that they're in. That's why they evolved, most likely. And so they have protective or therapeutic or disease preventive properties. You can get along eating, what's that stuff? Soylent, as your complete diet. You don't have to have any phytochemicals. That's sort of the definition. But you're better off if you do, and you'll probably live longer and happier and have better skin and have longer hair and whatever else you want if you get a lot of phytochemicals in your diet. So as I mentioned, we've coined with Tom Kensler, who was here for many, many years, coined the term green prevention. I would hope that the fact that we're talking about green plants and prevention of chronic diseases, that it makes sense that they come together. Pat, did you have a question? Just stretching. Okay. I thought maybe you were saying, hooray, green prevention. Okay. So we like this term. Pat likes it too, I think. I'm not sure. I never asked you. Do you like it, Pat? Okay. Pat Egner has been an integral part of a lot of our studies in China, which I won't talk about. And by the way, because of the department that we're broadcasting from, I've deliberately avoided talking about any of the studies that those of you in this department, what's it called now, EHE, are involved or have been involved in over the years. But this was certainly a big part of the work we did. I'll mention them in passing, but I won't dwell on them. Green chemo prevention, or frugal medicine as we call it, is the mechanism, just simply the mechanism-driven use of dietary means to deliver chemoprotective phytochemicals. We think it's realistic. We think it can serve the rich and the poor alike, and that it's economically sustainable. In the populations where these non-communicable diseases or chronic illnesses are becoming more and more huge problems, and certainly will into the future, suggesting or trying to persuade someone that they should or could switch their diet a little bit, tweak their diet and eat this plant instead of this one, et cetera, is at least within the realm of possibility, whereas buying courses of antibiotics ain't. I mean, there are broad swaths of the world, as you all know, where you just can't anticipate drugging someone with pharmaceuticals of various sorts to prevent a disease that they may be at risk of, enhanced risk of, or may never have to worry about. So we like green chemo prevention from a global or public health perspective. In terms of evidence that it works, there's overwhelming evidence I won't go into here. You probably, if you're a student, you've probably talked about it or will talk about it in classes. But evidence that high fruit and vegetable consumption reduces or can reduce the risk of a variety of chronic diseases. And we and others, many others now, have obtained what we think is overwhelming evidence that a variety of edible plants contain inducers of the so-called phase 2 or cytoprotective enzyme response in people. And we'll talk a bit more about this. So I mentioned Paul Talalay, who hired me here. He, I would say more than just about anybody, is responsible for coining this term and defining sort of what goes into chemoprotection. He has pointed out, and we've drunk the Kool-Aid, and most of us who follow in his footsteps repeat this mantra, that all aerobic cells are constantly stressed by reactive oxygen intermediates, of course, toxic DNA damaging carcinogens, inflammation, infection, radiation, a lot of UV radiation, that can lead to cancer and chronic degenerative diseases. Cells contain certain protective or so-called phase 2 enzymes, phase 2 genes Most of these are Nrf2 dependent, and we'll talk about that in just a minute. They normally don't operate at maximal capacity, but they can be induced by a variety of pharmaceuticals, a variety of small molecules, and by a number of dietary phytochemicals. And that third point is that upregulating these enzymes, a key feature of which is enhancing or producing higher levels of the main cellular oxidant glutathione, is highly protective and reduces the risk of cancers and a variety of chronic degenerative diseases. In cartoon form, this is Tom Kensler and I put this together. It's essentially, we're saying there are a lot of insults that ultimately can or could lead to chronic diseases. There is an alphabet soup of effectors, of transcription factors, et cetera, et cetera. I've just listed 3 of them for reasons that will become obvious. Exercise, environment, all the fun stuff you can get in excess and can enhance or perhaps reduce, well, I wouldn't say sunlight. Yeah, I would. Can possibly reduce the risk of chronic disease, but certainly we know that diet and the phytochemicals contained in what you eat can modify your risk for cancer. So why are certain vegetables especially protective? Well, we've zeroed in on the glucosinolates from cruciferous vegetables. By way of definition, well, I have the following slide sort of defines glucosinolates and isothiocyanates. I've told you about these enzymes very briefly, and sulforaphane is the prototypical isothiocyanate, which is in fact found in broccoli. But what actually happens is that in the intact vegetable, broccoli, for example, because that's what we've spent the last 20 or 25 years working on, broccoli has this compound called glucoraphanin. It is relatively stable, water-soluble, heat-stable, non-reactive. It just hangs out there. Remember, though, plants are mostly sessile. They sit still in one place, or they stand, or they're rooted in place, shall we say. And they can't run away, and they can't pick up guns and knives and fight off attackers. Attackers being you that are going to eat those plants, or animals that are foraging, or fungi or bacteria. So what they, of course, have to do is create a chemical arsenal to scare them off, make them reject the bitter taste, et cetera, et cetera. Glucoraphanin is pretty benign, though, and it sits in a bag, in a vacuole, in plant cells. And when predators, like you or your animal friends, chew on the plant, or your fungal or bacterial friends, when you chew on the plant, macerate the leaf, break the leaf, you release an enzyme that's membrane bound, and this enzyme is called myrosinase. It's in probably all of the same cells where glucosinolates occur, but it can't touch this molecule, its substrate, until and unless you bust open the plant cell and allow the juices to mix, if you will. I'm being deliberately very, very scientific here. When you do that, there's an enzymatic reaction, then a spontaneous rearrangement, and you get the formation of sulforaphane, which is an isothiocyanate, very, very reactive, heat sensitive, not very water soluble, or not as water soluble as its precursor. And because it's very reactive, it does a lot of things in human cells, and we'll talk about that. I should back up one. Before I put the X on it, you all do this reaction. I talk about this in my class in the late spring, and everybody's got a different example, but I like the example of a radish. When you chew on a red radish, it's cool and crunchy, and within a matter of seconds, 20 seconds, 30 seconds, what happens? If you're really sensitive and it's a hot radish, your nose starts to run, your eyes may start to water. It's a lacrimator. That's because you're doing this reaction. You're the big bad predator. You're breaking open plant cells. You're releasing myrosinase and allowing it to react with glucoraphanin. And this reaction obviously then happens very quickly. Broccoli has, the compound in broccoli is relatively mild, but it does give that distinctive taste that you either like or don't like. Radishes have an almost identical compound that is quite more of a lacrimator, makes your eyes tear. Why did I have the X there? Does anybody want to tell me why I had the X there? No? Most people don't cook red radishes, but for example, daikon, the Japanese or Asian variety of radishes, is very, very often cooked. When you cook a vegetable, you denature the protein, you knock out active enzymes, just as you do when you cook an egg and denature the white. You precipitate, you denature the protein. So when you cook cruciferous vegetables, you're knocking this out, and then you've got to count on your gut microflora. And it turns out, and Kitty and Chris and Wah in the back of the room there, who've worked with me on this for many, many years, have participated, we've all participated in studies where we've looked at either on ourselves or on volunteers, the activity of the gut microflora. And so we know that everybody we've looked at here in China, hospitalized, healthy, not so healthy, all have this enzyme in their gut. But it's at very different levels, very different activities, and we're not really sure which critters in this gut microbiome have the lion's share of the activity. All this is sort of in the works, and we and others are interested in it, of course. But we've shown, for example, in a small clinical trial that if you ablate the gut microbiome, if you give a fleet enema and a preoperative course of antibiotics to a volunteer, you completely and then feed them just glucoraphanin without myrosinase, you completely wipe out their ability to do this conversion. I'll share some studies with you that sort of address that. And then by repopulation of their gut microbiome over the next 2 or 3 or 4 weeks, they regain whatever ability they had to hydrolyze these compounds. We can call it a pro-drug and drug situation. For those of you who are human biologists or ecologists or environmental scientists and have never seen a plant before, I sort of doubt there are anybody who doesn't know what cruciferous vegetables is, but the ones that are most commonly found in all supermarkets are here: cauliflower, cabbage, Brussels sprouts, broccoli, radishes. Maybe less well known to some people are things like mustard greens, canola, rutabaga, Chinese cabbage, etc., arugula. So it's a huge family. There are upwards of 500, maybe 600 members of this family, and they all have this system of glucosinolate, an enzyme, and an isothiocyanate that's, we have looked at many of them, probably not all of them, certainly not all of them, but they all have some activity inducing the phase 2 response or the protective enzymes. So mentioned sulforaphane. Over the years, I think we feel very comfortable saying it's the most potent, or certainly one of the most potent naturally occurring inducers of these protective enzymes. It's found in very high levels in broccoli sprouts. We showed this a number of years ago, much higher levels than in the broccoli you normally see at the supermarket. Therefore, we've focused on broccoli sprouts for many of our experiments, but it's the same stuff as you find in broccoli in the supermarket. And it has a lot of modes of action. I mentioned the Nrf2 pathway, and I'm going to talk to you about a few others. But first, I just do want to say something additional about the Nrf2 pathway. I think, and this is my personal prejudice, but I think that after the isolation of this compound in the early 90s by Paul Talalay and his doctoral student, Gary Posner over at Homewood at the time, really great interest developed in this molecule. And I think it would have probably or perhaps just been another interesting phytochemical, and there are very, very many of them, and they all have different modes of action, had it not been for the fact that at the same time, they and a small group of people around the world mostly here at Hopkins actually, and in Tsukuba, Japan, were looking for the signal molecule, the transcription factor. No one knew exactly what it was at the time, by which this acted. So how does this exert its effect? Well, it turns out, and this was discovered in the early 90s also, that this Nrf2 pathway is one of the main places where the rubber meets the road, so to speak. So in this department, Biswal and certainly Talalay, Albena Dinkova-Kostova, Masa Yamamoto, who has a adjunct appointment in biochemical and molecular biology, and certainly Tom Kensler, a small group of people really put this pathway on the map. And a lot is known about it now, and a huge amount of work continues to go on identifying the ins and outs of this pathway. This is self-explanatory. I'm going to try to speed up because I always have more slides than I can get to. Everybody that I know knows that. Everybody that I've taught in a class knows that. So try to move a little faster and skip a few things. Sulphoraphane, Hua Lu and I did a review, and this is a figure from that, this is part of a figure actually from that paper, trying to just in very simplistic fashion summarize what happens in this Nrf2 pathway. In brief, molecules like sulphoraphane bind to sulfhydryl groups, reactive sulfhydryl groups, or sulfhydryl groups on reactive cysteine moieties on a dimerized chaperone protein called KEAP1. KEAP1 is tethered in the cytoplasm of cells, mammalian cells, and others, and it holds, again, very simplistically, it holds or keeps this transcription factor, Nrf2, in the cytoplasm. When sulphoraphane or other compounds bind to these cysteines, Nrf2 is released, and instead of being ubiquitinated and degraded, as it does cyclically, it goes to the nucleus. It binds to the antioxidant response element, and it upregulates the production of a whole series of so-called phase 2 enzymes. Some of them are listed here, but there are very many of them. And our colleague Albena, who's now in Scotland, has told me, and this is a sloppy number, but that something between 3 and 6%, I think, of the cellular proteins are actually transcribed or coded for by genes that the Nrf2 activates. Sharada, is that am I getting that number close? Where would you put it? A little, something like 5%? Okay, Sharada's recently joined our group too, and she comes from the lab in Scotland that I'm just talking about. So when that happens, when Nrf2 upregulates these protective enzymes, we call sulphoraphane, I didn't phrase that quite right, I suppose, but we call sulphoraphane, or SF abbreviated here, an indirect antioxidant. And the reason we call it an indirect antioxidant is because what it does is it, through the pathway I just told you about, upregulates a variety of antioxidant enzymes. What happens to enzymes when they're just hanging around? Do they go away right away? No. They have half-lives that are highly variable, but in the case of many of them, 2-3 days, something like that. So that these enzymes then can hang around and do their reactive oxygen scavenging or their other activities to reduce oxidative stress in cells for quite a while. What actually happens in the body, whether it be plasma or the urine or whole blood, when we do the pharmacokinetics, this represents data from 10 years ago now, but and this is a log scale on the left-hand side. The peak of sulphoraphane and its metabolites occurs very, very quickly, within a few hours, and then it disappears. Why? Because it's a reactive compound. It gets into cells. It's a xenobiotic. It's a foreign compound, and cells actually conjugate it with glutathione and get rid of it. And Pat Egner, who I was joking with earlier, has done a lot of mass spectroscopy, quantifying and identifying the compounds and the pharmacokinetics of excretion. But the body gets rid of this compound fast. However, in the process, it upregulates protective enzymes, including, I should say, I mentioned glutathione, which is what ultimately conjugates sulforaphane and gets rid of it. So it's dumped back out of the cell after it's already upregulated these. If you look at something like vitamin C, which is a direct antioxidant, it has similar pharmacokinetics, really. It peaks very quickly, and then it disappears. But it doesn't have the same enzyme activating or enzyme upregulating activity. It's sacrificial, and it goes away. So it doesn't last, and we and others call it a direct antioxidant. I say and others because there's actually a quite useful review that just came out that we have nothing to do with, but a number of people are starting to adopt this nomenclature. And then there are compounds like lutein, which are not a great direct antioxidant, not a great inducer of the phase 2 response, but they do both things. So it's sort of intermediate. Because of this broad protection by sulforaphane, we see, and this is a review by colleagues at Dartmouth from a few years ago, we see actually broad spread protection in a variety of organ systems. And this is documented by now a burgeoning literature in really all of these systems, model systems, not all clinical trials. But before I leave the pathways, I should just add a little complexity and tell you that there mitochondrial dysfunction, inhibition of the NF-kappa B pathway, the pro-inflammatory pathway, the heat shock response, all of these are pathways that we know are also impacted by sulforaphane and the isothiocyanates. And thus, if we sort of follow the pathways to the diseases, we have to consider these also. So we've done just that, we and others. We've been involved here at Hopkins in the School of Medicine, the School of Hygiene, in a number of prevention trials. Of course, when you're looking at cancer prevention, you're not looking at cancer as an endpoint. Usually you're looking at biomarkers of cancer in at-risk populations. And so, again, with a number of colleagues here and elsewhere, this China is actually, these are the studies Tom Kensler and Pat Egner and others here have spent much time on, and we've worked with them. But broccoli sprouts or extracts of them were used in all of these. If you're after more instant gratification and quick endpoints, certainly there are many non-cancer endpoints that one can look at, such as erythema or reddening of the skin, and a variety of other conditions. And we've been involved in trials on all of these, or there are a couple that are pending that haven't been funded yet. But so I present this, I can share this presentation with anybody who'd like it. I present this just to call attention to the fact that there are now really a large number of published peer-reviewed trials showing the use of broccoli sprout extracts or sulforaphane in people, clinical trials, with Nrf2 related outcomes. Albana Dinkova-Kostova and Tom Kensler and I have summarized this most recently in a paper that's listed here. And so these are a variety of disease conditions, in all of which there's an Nrf2 link. And then a second list in which there are a variety of pharmacokinetic and pharmacodynamic outcomes that have been evaluated, all non-Nrf2 related or not solely Nrf2 related. This is just to highlight the fact that Nrf2 isn't the only actor in town. There are very many pathways, it turns out, including we showed years ago a selective antibiotic effect against Helicobacter. Don't have time to dwell on these. I'd like to highlight 3 or 4. We'll probably only get to 3 of these studies and just dive into them a little bit more deeply. I need a time check. I got a time check. Okay, we have a few more minutes. We've done a lot of work on bioavailability. Frankly, we have a small group, and there are some complex studies that it's just not feasible to attempt. Bioavailability has been an interesting road to go down because we've been able to look at individuals, use individuals as their own controls, and challenge them with either glucoraphanin, the precursor, or with sulforaphane, or with a combination of glucoraphanin and myrosinase. And I'd like to just share a little bit of that with you, because we think ultimately, as one makes recommendations from a public health perspective, a disease prevention perspective, you've got to pay attention to how much is actually getting into people. Whether it's 10% of the dose or 100% of the dose, and the range is actually pretty much that wide. So we make this broccoli sprout extract. Kitty and Chris and I have made this for years now, and it's extremely time consuming, and I'm going to come back to that because it's a very important point to talk about from the perspective of people at a university doing these sorts of trials. So we make these broccoli sprout extracts, and if we add myrosinase and convert the extract to what I'll call the sulforaphane form, it's rich in sulforaphane. We know how much of it's in there because we measure it by HPLC or mass spec. By and large, so each dot represents an individual person's 24-hour excretion of sulforaphane and its metabolites. By and large, we get 70% or so back, but there's great person-to-person variability. The first bar is hospitalized subjects in this country, in Baltimore. The second bar is free living subjects in China. The dotted lines they're a fudge factor to account for the fact that these weren't full 24-hour urines. But the gist of it is a lot of the sulforaphane comes back. If we give glucoraphanin, not so much. And again, a lot of variability. When we first started doing these studies, we attributed the variability, the great variability here, for example, to the fact that people have different levels of myrosinase in their gut, which is certainly true, but that doesn't account for the high variability in cohorts like this. We then got the bright idea that we should add active myrosinase to these doses so that we could eliminate the variability person to person. Well, it turns out, everybody thinks they're really smart designing experiments. It was a good experiment, and we're glad we did it because what it showed is that we sort of split the difference, and we got an intermediate level, about 35 or 40% of bioavailability of ultimately sulforaphane. But it really didn't reduce the variability, and I think there's another slide sort of summarizing all the data points we have on that. So variability is still high, but the bar got moved up. And then, and this is all published in the last few years. So I told you I wanted to come back to the fact that it's unwieldy and difficult to do this in a research university, especially a hospital setting. We were literally going back and forth to the West Coast to the biggest freeze-drying plant in the world to make and freeze-dry these extracts and bring them back here and testing for titer, testing for bacterial load if there was any or whether there was any. And that was just the tip of the iceberg. So we were my group was spending about half of our time doing this for years to supply other clinical trials and trials that we were doing ourselves. Essentially, we had to shoulder all of these tasks or responsibilities, efficacy, which we'll talk about, cost, delivery, taste, clinician buy-in. It's not like a supplement or a drug where the pharmaceutical company or the supplement company's already gone out and persuaded physicians or healthcare folks to deliver it. And then things like logistics and distribution, food safety is a huge problem that we had to shoulder, and all the regulatory hurdles. I mention this because what we ultimately wound up doing is throwing our hands up and saying, we give up, can't do this anymore. And by that time, by 2015, of course, we had a lot of collaborators and friends who wanted these materials. So we actually went to a number of supplement companies, and one of them And I see your representative, Grace Kornblatt, is here from Nutramax, which is the company we went to that agreed to help us with this. We actually had a number of companies that expressed interest, but they agreed to help us make, I believe that already made this product, but to use a supplement that they took the responsibility for safety, microbial counts, quality control, et cetera, et cetera. They took that responsibility. The other important fact, and this I can't stress enough, is that if you do a clinical trial, if you as a university researcher do a clinical trial with something like what we had, a calibrated extract of broccoli sprouts, and you're successful, I mean, it's nice to think about success. Not all trials end in a big disaster. So if you're successful, what do you do when your subjects or their relatives or their friends or the parents of autistic kids, and we'll get to this in a minute, come to you and say, whoa, that worked. My kid got better, or I got better. I need more of that stuff that was in the trial. I need more of the broccoli. This is something we got all the time. Well, I'm a university lab. We make it in the back room cleanly, et cetera, et cetera. We can't sell it, and we can't endorse other products really either. So there's a real ethical, moral dilemma, because the whole point you're doing, the whole reason you're doing these studies is to hopefully have an outcome that will ultimately help people. What if you do help people, even if it's not the subject of 7 randomized trials, multi-center trials? You may well have subjects who come to you, former subjects, I should say, who come to you and say, Doc, it's irresponsible. In my case, our case, it's Brock Doc. You can't do that. You have to tell me what to take. So for many years, I resisted touching the supplement companies because there's conflict of interest. There are a lot of issues associated with dealing with companies, pharmaceutical companies, et cetera. But ultimately, we decided to throw up our hands and say, help us, please. And as I say, a company called Nutramax and some others did. But we first had an agreement with Nutramax, a memo of understanding. And essentially, and I am very grateful to them for this, essentially they agreed to the following: that they'd provide sufficient supplement for our use and that of our collaborators. So if we needed it, we'd get it. That allow us to inspect and test their formulations ad nauseum, which we do. They would also create placebos, tablets or pills. That was very important from the perspective of a clinical study. They would retain samples for long-term stability testing. This stuff is always made in lots, batch-to-batch variability. You got to hold on to it and test it again and again and again over time, and mimic the conditions that the material is used in the field. They agreed not to use the name of Johns Hopkins in marketing, which is very important to my being able to get buy-in by the administration here, of course. And they agreed to, and this is probably the most important and most odious task, and that's to assist not only our trials, but our collaborators that use this by timely provision of information for both IRB submissions and IND submissions to the FDA if required. That's a huge hurdle, and they have really stepped up to take care of this. And it's allowed us to use this material in a number of clinical trials now. I mentioned some other products. There's one that's rich in sulforaphane only. Unfortunately, it's not sold in this country, and because of that, this whole paradigm of what do you say to your subjects if you actually are successful becomes a little bit more of a problem. You can't say go to France and buy this stuff and bring it back on the plane. That doesn't work very well. Nicole, could you give me a time check?
I have about 20 minutes.
Okay, thanks. So these are the products that we've been using. We're actually using a product by another company called Thorne in a dermal test, dermal clinical trial, skin trial, so to speak. That's the French product, which is not available, and that's the Avmacol, the product that we've, as I say, used in a number of trials. Okay, moving right along. Let's talk about autism or or ASD, autism spectrum disorder, for a few minutes. I told you that broad protection by sulforaphane is possible. Various organ systems, the brain was at the top of the page. I didn't tell you, but you should know that sulforaphane crosses the blood-brain barrier, which we think, of course, is important in neurodegenerative or neurodevelopmental conditions. And sulforaphane and other Nrf2 active compounds have been shown to have promise in a variety of such conditions, including autism, schizophrenia, Parkinson's, et cetera, et cetera, et cetera. All of this really in animal model systems or even in cell culture. I will briefly share with you what's been happening in autism and in schizophrenia. The autism story. Despite extensive investigation into the causes, there aren't any cures. There are a few medical options for treatment of some of the most distressing symptoms. We know that there are biochemical abnormalities that are related to metabolism, and we know that the cells of people with autism frequently have high levels of oxidative stress. But there aren't any drugs for treatment. We also know, and I'm sure most of you are aware, there are certainly genetic causes. There are mutations that are associated with the prevalence or penetrance of autism in families. So there are genetic and epigenetic links and microbiome links, which are quite interesting. I wish we had time to talk about them. And there are environmental toxins and stressors which may be involved in the causative pathway. Now, I'm not espousing the Robert F. Kennedy and Jenny McCarthy approach that you shouldn't vaccinate your children at all, but certainly you can't discount the potential environmental factors. But layered on top of that, or in this slide beneath that, there are a number of mechanistic pathways or mechanisms that appear to be involved in the pathophysiology of autism. None of it proven, but a lot of associative work. And we know that all of these, or certainly 4 of the 5, can be impacted by sulforaphane. We also know, and again, Hua sitting in the back, and I and Paul Talley reviewed sort of what might be good biomarkers for autism that are easily accessible in a population, and that by coincidence might be moved or impacted by dietary treatment with something like sulforaphane. These are the 5 that bubble up to the top all the time. And you'll notice, I think the next slide, I showed you this slide a few minutes ago. What do you know? Mitochondrial redox. Oh yeah, mitochondrial redox, KEAP1, Nrf2, NF-kappa B, and heat shock. Basically, all of these pathways that we know from cell culture and animal and human studies that sulforaphane is so effective in impacting are involved one way or another in autism. We and others think. There's a lot of data in cell culture and animal systems showing that sulforaphane is effective. This isn't in people with autism, though. For example, it increases mitochondrial mass in cell cultures, increases mRNA expression for the heat shock protein 70. And on and on and on. This is just one. This happens to be work that was done by one of our collaborators, but many people, Sharada in the back and others have done work showing these effects in culture and in animals, and with knockout animals. I mentioned heat shock. So there's a link here. It sounds like I'm not getting to it, but intriguingly, about half of parents of autistic kids report that their children's autistic behavior improves substantially during episodes of febrile illness, and then reverts to baseline when the fever is gone. Our colleague, Andrew Zimmerman, who was at Hopkins and then went to Harvard and now is at UMass Medical Center, confirmed these anecdotal observations. Although a mechanism wasn't identified, certainly the suggestion that the heat shock response might be involved was there and was stressed. And in this paper, which they published 10 years ago, essentially, I won't dwell on the data, but essentially they showed that about half of the kids responded to fever by having a relief, or their symptoms got better, and then reverted when they went when the fever resolved. Here's their data. I better try to speed up. They also, Zimmerman was one of the co-authors of this paper. They also looked at things like cerebrospinal fluid. This study, I believe, was used age-matched control, neurodevelopmentally normal kids. But they found, dramatic increases in things like interferon gamma and some other potential markers in the cerebrospinal fluid. The data is not nearly as compelling, I'd say, in blood or urine or other metabolic compartments. But with all of these hints, Zimmerman came to us a number of years ago and said, let's do a clinical trial. You and others know that sulforaphane impacts the heat shock pathway. We think that the heat shock pathway is involved in this fever response. Let's see if kids get any better if we feed them sulforaphane, broccoli sprout extracts. We published this in 2014. Very briefly, it involved an 18-week intervention followed by a 4-week washout where everybody went on placebo. There were blood chemistries, urines collected, and behavioral analyses at various points. The subjects were split between broccoli sprout extracts and placebo 2-to-1, so there were actually more broccoli sprout takers than placebo takers. And we saw that by a variety of scales of standard measures of behavior, one being the aberrant behavior checklist and another being a social responsiveness scale, we saw that those on sulforaphane, 26 versus 14 on placebo, had a significant and substantial movement in their scores. This lower is better in this case, is an improvement. And when, very interestingly, and we were, of course, we weren't actually prepared to see this, but after the 4-week washout, their behaviors reverted more closely to baseline, significantly in one case and just barely in another. 10, thanks. When we looked at the individual subscales, and again, this is Zimmerman and his colleagues at Harvard that were doing this, but there was improvement in a number of the subscales, characteristics of autism, and resolution of at least some of them in the washout period. So overall, out of 26 that were on sulforaphane, 17 of them improved by objective measures by these scales. And we then did something, we're in the process of doing something that's a little bit strange. We followed up those subjects years later, 3 to 5 years after the original intervention. We followed them up because they were, all of those who were said to have improved on sulforaphane were absolutely adamant about continuing the intervention. As I mentioned, we could do nothing. This was stuff that was made at Johns Hopkins by us. But what our colleague Andy Zimmerman and his team did is they pointed them to the few, and at that time there were very few supplements, nutritional supplements, that we had tested that we knew had at least, I'm talking about sulforaphane or glucoraphanin, of course, had what they said they had in them. There are a lot of dietary supplements that are utter trash, they're garbage, that have filler, that don't have what they say they have in them. And we've tested a lot of them, and we've actually toyed with the idea of posting on our website a sort of outing of some of the lousy ones, and the Hopkins lawyers don't really like that too much for reasons that you might not be surprised to hear, but we might still try to do it, I don't know. So we've tested a lot of them, but the recommendation from Zimmerman was, okay, here are 3 or 4 that we know should be decent. If you want to go out on the market and buy them and give them to your kids, fine. Almost everybody did, and we knew that. And when we followed up with them, as I say, years later, this was last summer, the final follow-up, we actually put together what I guess you would call a case series, sort of a modified or a squirrely case series, because it's not well controlled. We went to those parents and interviewed them and got the stories that they were willing to offer to us. This has now been accepted and is in press. And so essentially of the 17 improved, we captured information from 14 of them and 2 that didn't improve. There was one kid who apparently after being on the intervention got tremendously better. I mean, I use these superlatives, but we have a little story from the parents. I mean, they couldn't go to the concerts. They couldn't go to the grocery store without trashing the store. They were violent. They were acting out and misbehaving, and now they're model citizens. This was all of these children are being seen by the team still. So it's been confirmed that indeed their behavior was radically improved. But so there's one that improved radically, a few that didn't continue on supplements, and there were 9 that are still taking some sort of a supplement and doing remarkably better in most cases. That study was supposed to include an examination of biomarkers, and it didn't. For various reasons, the bloods were collected, biomarkers weren't evaluated. 5 minutes? 7 minutes, thank you. Including question time. Oh, well, you didn't tell me that. Okay. We will talk faster. So we now have 5 studies in progress. A couple of them actually, one of them finished, in which we are massively collecting data for biomarkers. And those of us in the Chemo Protection Center here are actually doing much of the biomarker work. So we know what's going to happen. As well as all of the behavioral and social and behavioral symptoms, which are being evaluated at the respective institutions. So, for example, and I should mention, they're all now using Avmacol, the commercial product being produced up the road in Maryland by Nutramax. We've tested all these study supplements, not study drugs. Some of these studies asked for and received INDs from the FDA. Others didn't. Go figure. It's the same disease. It's the same supplement. It's the same label, the same bottle. This is one of those very gray areas where we are going to enter a very difficult period, because think about this. If a dietary supplement or a food is shown unequivocally, let's say there are 100 million studies and they're all just fabulously positive. If they're shown to affect a disease, symptoms of a disease, then what do you call it? Do you call it a drug? Well, at this moment, the FDA wants to call it a drug. I would suspect there'd be an uproar about that, but it's a question to ponder. So at UCSF, have a not placebo-controlled trial. The paper's in peer review now. We looked at urinary metabolites, metabolomics with this commercial company, identified a number of urinary metabolites that correlate with clinical improvements in children that are treated for autism. So this is exciting. The paper's not accepted yet, but hopefully will be soon. Another one is a large Department of Defense funded study that we're doing with Andy Zimmerman, who's now at UMass Medical Center. He's our collaborator on the published paper. And in this study, we're looking at a much younger age group than the previous study, 3 to 12 years, with the logic that this is the critical period to catch kids, and if you're going to be successful intervening, start early. So 23 of 50 subjects were completed. And he presented, I was a little surprised, but he presented at a public meeting that he and I both were invited to speak at in July, presented the initial data just in short form, and that is that in the intervention phase of the study, about 35% of the kids responded positively to whatever. They were either one-to-one on placebo or Avmacol. And then there's an open label phase after that in which everybody was switched to the supplement with sulforaphane. And 75% of those kids responded positively. So we're ecstatic that it seems to be replicating the previous study. And as I say, we're getting ready to start processing biomarkers in this study. Really quickly then, another study on schizophrenia, a set of studies on schizophrenia that are underway, sort of similar logic. They're also using a over-the-counter supplement. One's at Shepherd Pratt with colleagues here at Hopkins participating. Another is in China, a quite large study with 180 subjects. I was hoping to have some preliminary data on that for you, but I don't. Because Fahey is incapable of having the right number of slides for the time provided and he gets carried away, we're going to skip a discussion of skin cancer, which we've done a lot of work on, and I'm going to fast forward here. I would just like to tell you that we have a study underway now in which, condensing a bunch of slides, we've done many studies over the last 10 years showing an effect of topically applied sulforaphane on skin as a protector against ultraviolet light-induced or full spectrum sunlight-induced erythema or reddening, sunburn, if you will. Those trials were very successful. We've also, in animals, showed that we can protect against chemically induced skin cancer, UV-induced skin cancer. But we've never used a dietary delivery of sulforaphane. We're now doing that with Siwan Kang and Anna Chen and others in dermatology here, people from Pierre Coulombe's lab. And this is the industry has coined this beauty from within, but in other words, you take something and it helps make your skin absolutely wonderful. And sunburn, deliberately administered sunburn is one of the metrics we're using. This trial actually uses curcumin, for which I deliberately don't give attribution. Curcumin solves everything, fixes everything, cures all diseases. Check this out. All pathways are inhibited or activated by curcumin. I have no idea where these came from. All pathways. I mean, it's so wonderful. And of course, a lot of companies are exploiting this. It's also very not bioavailable, although again, there are delivery systems where that seems to have been improved. So we are using curcumin and sulforaphane, and the question is, is there synergistic reduction in redness at all between them? Because we know they operate by both similar and mutually exclusive pathways against inflammation. We expose, hopefully unexposed skin. Well, for those who are nudists, I don't mean to offend, but most people keep that part of their anatomy covered most of the time, and so it's a good substrate for sunburns. Using presently, because dermatologists have us sort of brainwashed, but using light-skinned people, Fitzgerald skin types 1, 2, and 3, because it's easiest to readily see the reddening, the burning. And we're giving them no intervention first, and then either glucoraphanin or curcumin or both. And we're taking biopsies. Each of those purple X's represents a skin punch biopsy, a deep skin punch biopsy. And we're not paying people a huge amount of money to get tortured like this. But we have a lot of volunteers coming through already. Wrap it up? Okay. Thank you, Nicole. So skin punch biopsies, this actually happens on both sides of the buttocks. So we have 8 skin punch biopsies. We hope to get high quality data out of this. I talked about INDs or not. For this trial, interestingly, the FDA actually said, yes, you require an IND for this. but no, we're not going to make you submit one. A completely two-faced schizoid response. So we did not have to get an IND, but we had to go through 9/10 of the process. I'd like to thank many people for recent funding. Most of our work isn't funded by the NIH. It's funded by, generous people, companies, groups. We have a lot of collaborators here and around the world over the years. We have a lot of wonderful people in the group, a lot of students who have contributed, and why do I keep punching this? I'd like to close by just challenging you to think about delivery of this stuff. I'm telling you about a food which we turned into a freeze-dried food, which then companies turned into a nutritional supplement, but you can go to the grocery store and buy broccoli sprouts or broccoli. So as this research goes forward, I used to think food companies should pay for it. None of them are stepping up. I used to think dietary supplement, nutritional supplement companies should pay for it. They should, but when you think about it, if the FDA turns what we're doing into a drug, why should they waste? Why should they spend their money on a product with a relatively low margin compared to pharmaceuticals to develop this base of data to then have it pulled out of their hands? There are no patents involved except for formulation, and we're talking about natural products. So pharmaceutical companies aren't interested. All they want to do is make better molecules, understandably, that are patentable, and forget about this category. Those are some email addresses and phone number. I'd love to hear from you. Love to share the presentation. Thank you. Sorry it took a while.
Hello. In the slide that when you mentioned the trial, randomized trial that was published, I believe, in the Global Advancements in Health and Medicine, when you said that the behavior has significant improvements, I just wanted to know if the supplement was taken in conjunction with other medications.
Yeah. Good question. So the paper that I referred to has not come out yet. It's been accepted, and it'll come out hopefully within a month or so. But yes, this was essentially patients who had been in our trial and then were completely disentangled from us, except that their kids were seeing psychiatrists and caregivers at either the Mass General or UMass Medical Center or a clinic, the Lurie Clinic. So they're all being seen, certainly. So there was knowledge of how they were getting along. But many kids with autism are on medications of one sort or another. So since we had, strictly speaking, we had no involvement with what they were, their care anymore. Yes, they were on whatever normal medications they were on. What we've done and what I believe published in the supplemental of the PNAS paper from the first trial is we indicated what medications people were on. Nobody felt comfortable telling parents of kids or young men in the case of the first study with autism to go off all their medications, whatever they may be. But the point is that there is no medication for autism. So there are a few meds that sometimes are given, or frequently, I guess, are given to kids to sort of reduce anxiety and repetitive behavior, and so you can damp down some of the symptoms, as I understand. I'm not a psychiatrist, but you can damp down some of the symptoms and perhaps control them. But there are side effects to all those medications too, especially for a developing brain. It's a great question. Hopefully that paper will be out soon. We've been waiting forever. As I say, it's a weird paper, so it was hard to get it reviewed. Other questions?
Thank you so much.